In this video, we're going to cover how a DNA strand goes to mRNA, which then is translated into a protein, and we're going to specifically worry about what does that protein look like at a various pH, because we know biological systems, whether that's within a cell, different compartments of a cell have different pHs, or just like in a, let's just say a human, certain parts of a human will have different pHs. Now, what you want to have out in front of you is either flip back in your notes packet or have your biochemistry reference sheet. Is you want your amino acid pKa values, okay, so something that looks like this, and then you want a list of amino acids. Now, if you go to college and you take biochemistry, the very first thing they're going to have you do is memorize your amino acids, and it seems like a daunting challenge, but just trust me, it's not as bad as it seems. There's definitely ways to memorize them. So, DNA is made up of base pairs. The ones that are made out of one ring or one cyclic structure would be pyrimidine. And then the ones that are bicyclic with two rings would be purines. Two rings. And when I took biology in high school, I remember learning the phrase pure as gold. And I don't know if that's something that's still said today, but that helped you see that the purines are adenine and guanine. Additionally, A and T pair up, and then G and C pair up when they hydrogen bond. Okay, so DNA gets transcribed into messenger RNA, which gets translated into a polypeptide, which is just a protein. And a protein is just amino acids linked together. I am not a biology teacher, so I'm not going to get into the 3' versus the 5' and how all of that is important in biology. I just want to strictly talk about the chemistry. Okay, so if I am a little bit vague as far as that, just understand that's not really my vantage point here. So we have codons, and if you take an AP Bio or you remember from biology, a codon is 3 nucleotides, and these codes tell you which amino acid comes next in your protein sequence. All proteins start with your start codon, which is going to code for methionine, and they're also going to end with a stop codon. Now, a stop codon does not give you an amino acid. It just tells it to stop. So once it reads the stop codon, it doesn't produce anything. Your protein is done. Now, when we go into messenger RNA, all of your thymines are going to be turned to uracils. Okay, so that's just something that happens. Again, not getting into the biology aspect of that, but that is going to be something that's important because we're going to see that our codons, our 3-letter sequences here, we're going to figure out what they code for as far as your amino acids. So that's why it's important to have your amino acid chart in front of you, just like even though I'm teaching this, I have mine in front of me. That way I know pHe. I mean, I'm pretty sure that that's going to be an amino acid that I can recognize right off the bat. But as I just am stumbling here, I thought it was going to be phenylalanine, but phenylalanine, actually it is phenylalanine, that's my bad. I might get a little bit mixed up, so that's why I want to have things in front of me. That would be the 3-letter abbreviation. Okay, so no shame in that whatsoever. So what we want to do is, let's just say we have this DNA sequence. There's a lot of ways that you can think about this, but I just like to take this and I'm going to repeat it here. And when I go into mRNA, the only thing that is changing is T's go to U's. Okay, so I'm going to get some white out here. This T is gone, 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 and this T is gone. And my T's are swapped with U's. Okay, now this is my messenger RNA, and I'm going to convert that messenger RNA sequence into a protein sequence via codons. So what I'm going to do is I'm going to break these into my codons, or my groups of 3. Let's hope I can do this well. Okay, and I know that my first codon is going to be AUG, and it should be a start codon. So when I go up here, I find AUG, and I see that it's going to be methionine. Okay, methionine is always going to be my first codon. Now I need GCC. So here's how I'm going to read this. I'm going to go, my first letter is G, my second letter is C, and so where those intersect, GCC, I'm going to have alanine. Okay, so it takes a little bit of practice to read this chart, but I find it really pretty straightforward once you do it a little bit. Okay, CUG, okay, CUG, leucine. UGG, okay, UGG, tryptophan, I believe, TRP. I could be wrong there. And then AUG again, and sometimes it's helpful to just check them off as we go, since we have a lot here. AUG is going to be methionine. CGC, that intersects here. CGC is going to be arginine. And CUC, is going to be leucine again. And then UGA, we have UG and then UGA. Yep, and that makes sense. We're going to have a stop codon. So a stop codon does not actually give you any sort of amino acid. Okay, so here's the thing that you're going to need a little bit of practice with, and it'll come with time, is we need to be able to draw our protein backbone. And so how this works is we have our amino group, NH, and I don't know how many H's are on that N just yet. And then I have one carbon, another carbon, and then NH, one carbon, another carbon, and that first carbon is going to be our alpha carbon, okay? And then on that second carbon, we're going to have a double bond with oxygen. Okay, so each alpha carbon, that is where your amino acid group chain is going to break off, and it's going to be different, but besides that, everything's going to be the same. So I need 1, 2, 3, 4, 5, 6, 7 alpha carbons. So right now I have 2. So one carbon, or oops. NH, one carbon, 2 carbons, NH, one carbon, 2 carbons, NH, oops. One carbon, 2 carbons, NH, First carbon is alpha. Second one is beta, but we're not worried about beta here. Alpha, beta. Let's see how many alphas I have. 1, 2, 3, 4, 5. Wait. 1, 2, 3, 4, 5. Yep. 1, 2, NH. 6, 1, 2. And a lot of people would say NH at the end. However, this is what's different. When you reach the end of your protein, it's not going to be an amino group. It's going to be carboxylic acid group. Okay? So we always have an amine group. We always have a carboxylic acid group. Okay, we're at a pH of 1. That's pretty acidic. So my guess is here we're going to have our H's, most of them on. So what I'm going to do is I'm just going to draw each side group for methionine, alanine, leucine, tryptophan, methionine, arginine, and leucine. So I'm going to do this first one here. We have methionine. I'm going to go all the way up, and I'm not going to do this for every single one, but I want to show you what I'm looking at. And I'm looking at this, and I'm looking at each amino acid. So there's methionine. It's 2 carbons, a sulfur, and then a methyl. Okay, so what will that look like here? One carbon, 2 carbons, a sulfur, and then a methyl group. When I go up to my pKa table for my amino acids, methionine does not have a side chain pKa, and that makes sense. The only H's off the side chain are alkane, and that would be 50. You're not going to have a biological system with a pH of 50. But we do have the amino group, and so what that means is this is going to be an NH3 group. I keep doing that. This pKa is 9.3. Okay? And what that means is at pHs above 9.3, this turns into NH2. We are not above 9.3, so that H stays put. Now we have alanine, so I'm not going to scroll up, but I'm looking at alanine on my helpful biochemistry sheet, and I see that alanine is just a methyl group. Alanine does not have a side chain pKa that is useful to us in a biological system, so that's all I draw for alanine. I draw leucine. Leucine is a carbon, then another carbon, and then off that carbon branches 2 methyl groups. Leucine does not have a biological pKa that's useful to us, so we don't need to take off or add any hydrogens. I've done methionine, alanine, leucine. TRP, which is tryptophan, and this one is always hard to draw, so just sort of be patient with it. Oops, and I've already drawn it wrong. That's how hard it is to draw. Okay, so we have something that looks like this, and then this one is like this. Okay, so we have tryptophan. Tryptophan, we don't need to worry about the carboxyl or the amino group because it's not at the end. It's in the middle of our chain. But we've got to check tryptophan to see if it has a side chain pKa, and it does not. Okay, so we're good on this one. That makes sense. This NH group is going to have a pKa of 38. You're not getting the an amino acid at a pH around 38. Okay, so we've done tryptophan. Methionine, we already know what methionine looks like. 1, 2 carbons, then S, then another one. No side chain pKa, so methionine is good. Arginine. Arginine's another weird one. We have 3 carbons, so 1, 2, 3, 1, 2, 3. And then we have an NH group. And then we have another carbon, that's double bonded to an NH2 positive group. And we have another NH2 group. Okay, arginine does have a side chain pKa, and arginine, this pKa of this NH2 group is 12.5. And what that means is that one of these hydrogens comes off if the pH is above 12.5. Our pH is at 1, so that H stays put. We don't need to take it off. We are going to do an example where the H's start to come off, but we're starting very slow. This takes a lot of practice to get this down. So now we're going to end with leucine. We've done leucine before. Leucine is carbon-carbon, and then they branch off into two methyls. However, on leucine, we're at the end, so we've got to figure out what the pKa of that carboxyl group is. For leucine, this pKa is 2.3. That means that at a pH greater than 2.3, this H comes off. What would that look like? Let's just change this and let's say this is 2.4 just for a quick second. That means that H has been removed and it would look something like that. Okay, that's not the case that we have here, but that's just where we're going. That's what these would look like. Okay, so this is what that protein looks like. Okay, let's do the second example. I'm going to go a little bit quicker as far as sequencing, and then I'll still slow down when I actually draw my protein. Okay, so DNA sequence, easy enough. All I'm going to do is change my T's to U's. Okay, and then I'm going to slice them by 3s. AUG is methionine. GCC is alanine, ALA. CUG is leucine, LEU. UGG is TRP, tryptophan. AUG we know is methionine, we just did that one. CGC is arginine. CUC is leucine. And then UGA, that must be our stop codon. So we have, again, 7 amino acids. We're at a pH of 2.3. And watch how I'm going to just structure this to come up with my protein. I know I'm going to have HN12NH, 12NH, 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 12NH. Now I gotta check how many alphas I need since I have 1, 2, 3, 4, 5, 6, 7 amino acids. I need 7 alpha carbons. 1, 2, 3, 4, 5, 6. So that means I'm gonna have to make this a bit smaller. No worries. And so on my last one, which I know I'm on my last one, it's not going to be NH, but it's going to be OH. And then on my beta carbons have a double bond to O. So methionine is going to be 1, 2, sulfur, methyl, and methionine does not have a side chain pKa, but the pKa of my amino group is going to be 9.3. So that means we're below that. If we're below it, the H stays on, so we have NH3. If we were at a pKa, or if we were at a pH of 9.4, this would be H2N. Okay, so that H would be removed. Okay, alanine, if we look at alanine is just a methyl group, no side chain pKa. Leucine, carbon-carbon, then they branch, no side chain pKa. Tryptophan, this looks very similar to the one we just had above. And it is. Okay, I should have noticed that sooner. Tryptophan is carbon, and then we start that, no side chain pKa, methionine, 2 carbons, sulfur, arginine is going to be 1, 2, 3 carbons, 1, 2, 3, and then an NH, then another carbon, double bonded to an NH2 that's positively charged, then NH2. And then leucine, we know that that is going to be carbon-carbon, then they branch. And leucine, this pKa is, let me look here, leucine is a pKa for the carboxyl group. pKa is 2.3. So since we are at 2.3, 50% of them will be on, 50% of them will be off. Let's just say we're at a pH of 2.4. That H will be off and will look like this. This NH2 group, I kind of went over that, but we've already talked about this. pKa is 12.5, so we're nowhere near that. Okay, let me just see if I'm at the same. Okay, I've got a different amino acid or protein here. I'm going to do this last one and then we'll call it quits. So again, I'll go a little bit faster, or I'll try to focus just on the things that are confusing to most people here. When we go to mRNA, our T's go away. We're replaced with U's. We break it up into groups of 3 called codons. And then we're going to use our codon table to translate this into an amino acid. We know we're going to start with methionine, GCC, GCC is alanine, CUG is leucine, UGG is tryptophan, AUG we know is methionine. Is this going to be the same one? I think it is. So what I'll do is I'll CGC, CG, C is arginine, and then CUC is leucine. Okay, so what I'm going to do is I'm going to copy this guy, and I'm going to show you all that is different here. We're just at a pH of 4.2. So nothing actually changes, okay? Because this pKa is 9.3, meaning that if I want to take off one of those H's, I have to get past 9.3. This pKa for one of these H's is 12.5, meaning I have to get past that pH in order to take off that H. So let's just change the problem, and let's just say we're at a pH of 10.0. Here's what changes. We've crossed that one, so that H was already taken off. We've just crossed this one, so this turns into NH2, and it's neutral. And we have not crossed this one, and so that's the only thing that changes there.